Hello viewers, in this video I am going to discuss about factors affecting enzymatic activity. That is, what are the various factors which can affect the activity of enzymes. But before that we should know that number one, enzymes are globular proteins. What are globular proteins? Globular proteins are those proteins in which the protein molecule is bended, folded or twisted in such a manner that it leads to the spherical structure or the globular structure. And the various factors which are responsible for this bending, folding or twisting of the protein molecules are number one, the ionic interactions, number two, the hydrogen bonding, number three, the disulfide linkages, and number four, the van der Waals forces of attraction. So these are the four interactions which causes the bending, folding or twisting of the protein molecule. And these interactions are in the side chains of the amino acids present in the protein chain, or you can say the peptide chain. So in the peptide chain, various amino acids are present. And in the side chain of the amino acid, there can be charges, the positive charges and the negative charges. So the interaction between these positive and negative charges will form the ionic interactions. While the van der Waals interactions, we know when the aromatic rings are there and then they interact, then this is the van der Waals interaction. Then the hydrogen bonding. The hydrogen bonding can be the hydrogen which is attached to an electronegative atom can form the hydrogen bonding with an electronegative atom, or there can be the disulfide linkage, the linkages between the two sulfurs. So these are the interactions which are responsible for the protein molecule to be folded, to be twisted or to be bended to give the globular structure. Okay, so this is that enzymes are globular proteins. This is all about the globular proteins. And then enzymes accelerate the rate of reaction by lowering the activation energy. So the rate of reaction in presence of enzyme is increased by many folds as enzymes decreases the activation energy. And as the activation energy is reduced, the reactants can easily overcome that barrier to form the product, and therefore the rate of reaction is increased. And third and the last point is that the basic enzyme catalyzed reaction is that the enzyme form complex with the substrate to give enzyme substrate complex, and this is formed when the substrate binds to the active site of the enzyme. So the substrate when binds to the active site of the enzyme results in the formation of enzyme substrate complex, and then the product is released and enzyme is regenerated and therefore can be used again. Now we can study the factors. So there are 5 factors which can affect the enzymatic activity. Number 1, temperature. Second is pH. Third is concentration of enzyme. Fourth is concentration of substrate. And fifth is presence of inhibitors. So now let's discuss all of them one by one. So first is the effect of temperature. Now if we study any reaction and see that what is the effect of temperature on the rate of reaction, then as the temperature increases, rate of reaction increases. So this is the shape of the graph, that on y-axis it is reaction rate and on x-axis it is temperature. So as temperature increases, rate of reaction also increases. Now if the reaction is taking place in presence of enzyme, then what will be the shape of the graph? That means how the increase in temperature will affect the rate of reaction in an enzyme catalyzed reaction. This is the shape. So as the temperature increases, rate of reaction initially increases. So why the rate of reaction increases? Because as temperature increases, kinetic energy of the molecules increases, and as the kinetic energy of the molecule increases, number of effective collision increases. So more and more products are formed and hence rate of reaction increases. So rate of reaction increases linearly, and then this is the optimum temperature. Optimum temperature means the temperature where the enzyme is having the maximum activity. So enzymatic activity is maximum at this temperature, and then if we increase the temperature beyond this, then rate of reaction decreases. And the reason for this is that as we increase the temperature, the enzyme gets denatured. What is denaturation of enzyme? I told you that enzymes are the globular proteins. So various interactions are present in the enzyme, or you can say in the protein, which leads to the globular structure of the enzyme. Okay. So, and I told you the 4 interactions which are present in the globular protein. So when the temperature increases, so the kinetic energy of the molecule increases, and therefore these interactions gets disturbed, because of which the enzyme gets denatured. And since the enzyme gets denatured, that means there is the loss of the conformational structure of the enzyme, and hence loss of the enzymatic activity. And therefore, as temperature increases beyond this optimum temperature, rate of reaction decreases, because now enzyme is not functional, not active to show the reaction or to act as a biocatalyst. Now, for example, optimum temperature for humans. For humans, optimum temperature is close to 37 degrees Celsius. So this temperature, optimum temperature for humans is 37 degrees Celsius. That is, we know that 98.4 Fahrenheit. That is a normal body temperature. Now, if a person has a fever, so a person who is having a fever, that means, and let's say the body temperature exceeds 40 degrees Celsius, that is 104 degrees Fahrenheit, then it will be a life-threatening situation, because it is a temperature at which the enzyme gets denatured. So there is a loss of functionality, or the enzyme is non-functional. So since enzymes are non-functional, and therefore this is a life-threatening situation, biological reactions will get stopped, because now the enzymes are not in the functional form. Okay. Now, the destroying effect of temperature can be used. We can use this, that as the temperature increases, after the optimum temperature, the enzyme gets denatured. We can use this destroying effect of temperature on bacterial enzymes. So enzymes which are present in the bacteria, and we want to destroy that bacteria, or we want to denature the enzymes which are present in the bacteria, so we can destroy that by increasing the temperature. And this is the thing which is used to sterilize the medical instruments. The medical instruments are sterilized in autoclaves. In autoclaves, the temperature is high. So at that high temperature, the bacterial enzymes become non-functional. And therefore, this destroying effect of temperature can be used to kill the enzyme, to kill the bacteria by denaturing the enzymes of bacteria. I hope I am clear to everyone. And now the second effect, effect of pH. pH of a solution has a great effect on enzyme activity. An enzyme behaves efficiently as a catalyst over a narrow range of pH. The optimum pH, again, optimum pH is the pH at which the enzyme exhibits maximum activity, is characteristic of a particular enzyme. That means each enzyme is having its own optimum pH value. That is, each enzyme is behaving or exhibiting maximum activity at a particular pH. Most of the enzymes exhibit optimal activity at pH value between 6 to 8. But there are few exceptions, like our stomach contains enzymes such as pepsin. And we know that in stomach, it is hydrochloric acid is present. Okay, so the medium is, the pH is low. And pepsin functions optimally at pH is equals to 2. Now, enzymes in a small intestine function at slightly basic pH. So, for example, optimal pH of trypsin is 9. So although most of the enzyme exhibit optimal activity at pH value between 6 to 8, but there are few exceptions. Now this is the shape of the plot. That if we draw the rate of reaction on y-axis and pH on x-axis, we get this bell-shaped curve. And what is the reason for that? That means here it is having the maximum activity, enzyme is having maximum activity here because here the rate of reaction is maximum, and if we lowers the pH or if we increases the pH, reactivity decreases. Okay. So reason is that at high or low pH value, the denaturation of enzyme takes place. Now, why the denaturation at high or low pH? Low pH means medium is acidic. If the medium is acidic, then what will happen? That the side chains of the amino acids which are having negative charge will get protonated. For example, if the side chain is having COO negative, carboxylate ion, and medium is acidic, then COO negative will exist as COOH, acid. It becomes neutral. And now, as I told you, that in the globular proteins, there is the ionic interaction present. So, ionic interaction will get disturbed because now there is no negative charge or less number of negative charge present because the negative charges are getting protonated, so they become neutral. So, the ionic interaction is disturbed. So, at low pH, the negative charge will get protonated and therefore they are not the molecule, the ion, the side chain become neutral, and because of this neutral side chain, it cannot form interaction with the positive side chain. Now, if the pH is high, as the pH is increased, so if the pH is high, what will happen? High pH means medium is basic. If the medium is basic, that means hydroxide ion is there. So, medium is basic. Then what will happen? Since the medium is basic, so it will abstract the proton of the positive charge. For example, ammonium ion, NH3 positive. So NH3, the side chain is having positive charge because of this NH3 positive. So the ammonium ion is having positive charge. Since the medium is basic, so it will abstract that proton and convert it NH3 positive into NH2 amino group. Again neutral. So the positive charge is getting the group, the side chain which is having positive charge will be deprotonated. Deprotonated means now it become neutral. So again ionic interaction is disturbed. So whether the pH is high or the pH is low, the ionic interaction is disturbed, which leads to the denaturation of enzyme, and therefore the activity is reduced. Not only the enzyme is affected by the pH, but the substrate is also affected by the pH. As in the enzymes, the side chains which are present can be protonated or deprotonated at different pH values. Same is applicable for the substrate. That means if substrate is having the negative charge, any group present which is having negative charge, so that will get protonated in the acidic medium. And if the substrate, let us take, is having the positive charge, NH3 positive, assume it is having NH3 positive charge, then the basic medium will abstract the proton. So it is deprotonation. So as in the case of enzyme, in the substrate also the groups present can be protonated or deprotonated. And this leads to the less efficient interaction between enzyme and substrate, or the interaction is even impossible. So the conformation of the enzyme is changed. The side chains present or the groups present on the substrate is also protonated or deprotonated. So the enzyme substrate complex is affected. The strength of the enzyme substrate complex, the concentration of the enzyme substrate complex is affected, and hence the enzymatic activity is affected. So I hope the effect of pH is clear to all of you. Now, the concentration of enzyme. So if we have to plot a graph between rate of reaction with increase in concentration of enzyme, then by keeping the concentration of substrate constant, that means if the concentration of substrate is constant and we keep on increasing the concentration of enzyme. So as the concentration of enzyme increases, rate of reaction increases. So this is the plot. So as the concentration of enzyme increases, rate of reaction also increases. So the greater is the enzyme concentration, the greater is the reaction rate. But we all know that enzymes are biocatalysts, that is, they are not consumed in the reactions where they are catalyzing the reaction. So they are not consumed in the reaction in which they catalyze, and therefore the cell usually keeps the number of enzymes low as compared to the number of substrate molecule. And the reason is that for the synthesizing and maintaining of the enzyme molecule, the cell has to pay the energy cost. Energy is required to synthesize and maintain the large number of enzyme molecules. And the cell can avoid this because the enzymes are in any way they are regenerated. So enzymes are not required in high concentration. And therefore, in general, the concentration of substrate in a reaction is much higher than that of the enzyme. Okay. And now the fourth. The fourth factor which affects the enzymatic activity is concentration of substrate. So this red plot indicates the concentration, the plot between the concentration of substrate and rate of reaction in absence of enzyme. So when no enzyme is there, so as the concentration of substrate is increased, rate of reaction also increase. So this is the plot. But if the reaction is taking place in the enzyme, then this is a shape. The rate of reaction first increases and then it attains the maximum value and it becomes constant. So this initially rate increases directly with the substrate concentration. That means as the substrate concentration increases, rate of reaction increases and then the rate approaches maximum. Now what is the reason behind this? As the concentration of enzyme is constant here. So here the concentration of enzyme is constant and we go on increasing the concentration of substrate. So as the substrate is added up, the enzyme and substrate form complex and which leads to the product. More as the enzyme substrate complex is formed, more will be the product formed and hence higher will be the rate of reaction. So as the concentration of substrate increases, rate of reaction increases. But there will be a point where it becomes constant or approaches to maximum. Now when the maximum velocity or the rate is reached, that is the point where all of the enzymes are saturated with the substrate. So enzyme substrate complex is maximum there and it cannot further increase because all enzymes are saturated with the substrate. They have formed complex with the substrate. And after that, if we increase the concentration of the substrate, the rate of reaction will not be affected. And this we can show as a evidence or a proof for the existence of enzyme substrate complex. How? At constant concentration of enzyme, the rate of reaction increases with increase in concentration of the substrate until a maximum velocity is reached. However, uncatalyzed reactions, that means the reactions taking place in absence of enzyme, do not show this saturation effect. That means in that case, rate of reaction increases as the concentration of the substrate increases. So at sufficiently high substrate concentration, the catalytic sites are filled and so rate of reaction reaches a maximum. Hence, this is the most general evidence for the existence of enzyme substrate complex. So I hope this point is clear to everyone. And now move to the fifth and the last factor, that is the presence of inhibitors. Now what are inhibitors? Inhibitors are the substances which inhibits or you can say reduces or sometimes stop the enzymatic activity. That means the rate of reaction is reduced by the inhibitors as they inhibit the activity of enzyme. Now, enzyme inhibition can be of two types. Number one, irreversible inhibition, and the second one, reversible inhibition. Now, as the name indicates, irreversible inhibition is the inhibition in which the inhibitor is bound very tightly to the enzyme either covalently or non-covalently, that its dissociation from the enzyme is very low. That means the enzyme and the inhibitor form complex, and this complex dissociation is very low. That means now the enzyme is not available for the substrate molecule to form the complex and hence give the product. And since this complex is strong, and therefore this inhibition is irreversible inhibition. This inhibition cannot be reverted back. While the reversible inhibition is that inhibition in which there is a rapid equilibrium between these two. That means enzyme react with inhibitor or enzyme form complex with inhibitor, enzyme inhibitor complex, and this enzyme inhibitor complex dissociates to give enzyme and inhibitor. So there is a rapid equilibrium. So this inhibition is a irreversible inhibition. Now, if you want to know more about the enzyme inhibition, you can watch my video on this topic, enzyme inhibition. So this is all about this video. So now today we have discussed that there are 5 factors which can affect the enzymatic activity. Number 1, temperature. Number 2, pH. Number 3, concentration of enzyme. Number 4, concentration of substrate. And number 5, presence of inhibitors. Thanks for watching.